Hello and welcome to week 7. This week we're going to talk about things at the chromosomal level, both eukaryotes as well as in bacteria. In chapter 5 we're going to talk a lot about linkage and how we know the distance between genes. One of the ways we can do this is by looking to see how often things like crossing over happen, especially if the genes are going to be on the same chromosome. In chapter 6 we're going to talk about bacteria, some of the different techniques we can do to study the genetics of bacteria, and we'll talk a lot about bacteriophages, which are a special type of virus that can infect bacteria. So first of all, what we're mostly going to be talking about this week is when genes are found together on the same chromosome. And what we know from when we talked about meiosis is that if genes are on the same chromosome, they're likely to segregate or come together. If they're on different chromosomes, they're likely to, or potentially can assort independently, but if they're on the same chromosome, they have a high chance or high probability of traveling together. So let's take a look at this, several different possibilities you can see here. In the first case here, this is going to be a case of independent assortment. You've got two different genes on two different pairs of chromosomes. You've got A here, copy is going to be the two copies here, and B. And you can see these are on two completely different chromosomes. This parent, A and B, again, two different chromosomes. So when these gametes form, you're going to have A on one of these chromosomes, B on a different chromosome, and so on. And the different possible combinations you can get is that you can get this copy from mom, this copy from mom, this copy from mom, this copy from dad, these both from dad, one from mom. And that's what we've talked about previously. Now, if these two genes are located on the same chromosome, A is here on this part of the chromosome, B is over here, A here and B here, when this undergoes meiosis and the gametes are going to be formed, if there's no exchange happening, if there's no crossing over happening, what you'd expect to see are 4 gametes, and they're all going to be like the parents. Now, we've talked previously about some genetic exchange of material. One of the ways that we can get this is through what we call crossing over. So we're going to call these genes, if they're found on the same chromosome, in this case, they're going to be linked. Okay, and we have a single exchange occurring between these two non-sister chromatids. So you can imagine these guys, they're forming a synapsis, and they're undergoing this crossing over event. So you have now 4 different possibilities still, but the variety that you can get is now increased. So this parent, this gamete you could get looks just like this parent, this one you would get would look just like this parent, and neither of these would have any crossing over. In the 2 that are crossed over though, instead of having these 2 from the parent, you have one from this parent and one from that parent. And you can see right here the demarcation where the crossing over event happened. And then the other one would be its counterpart here. You can see again where that crossing over event has happened. Okay, so think about if you've got, say, 2 hypothetical recessive autosomal genes, call them A and B. What would happen if you took a heterozygote for those and you test crossed it to your double homozygous mutant? What type of phenotypic ratio would you expect if these 2 genes are located on separate chromosomes? What would you expect if A and B are linked on the same chromosome but so far apart that a crossover almost always occurs between them? And lastly, if A and B are linked on the same chromosome, but they're very close together, and so in this case you may never get a crossover between those 2 genes. There's just not enough space for something like that to happen. So I'll let you go ahead and pause it now and think about the answers to these. Okay, so in the first case, you would expect to see a 1-to-1 to 1-to-1 ratio. And this is what we've discussed before when we've done our typical Mendelian genetics. And you can draw a Punnett square for this, a 2-trait cross, capital A, basically this individual genotype times somebody who is this genotype what ratio of offspring would you expect to get, and you would indeed expect to get a 1-to-1 to 1-to-1 ratio of these 4 genotypes. Okay. Now, if A and B are linked on the same chromosome, but they are very far apart and a crossover almost always occurs between them, you're actually still going to get that same ratio. Okay? However, if A and B are linked on the same chromosome, but they're so close together that you're almost never going to get that crossover occurring, primarily they're going to look like the parents. Okay? These are going to stick together here, these are going to stick together here. Okay. This crossing over events basically served as the basis of determining how far apart genes were when they're trying to map chromosomes of different organisms. And one of the most important ones that they studied for this was Drosophila. And Thomas Morgan was the lead scientist on many of these studies. He just studied Drosophila mutations on the X chromosome. He looked at a number of different traits, and what he concluded was that these linked genes must all be arranged in a linear sequence along this chromosome. And you're actually able to calculate, again, Drosophila, you're able to cross any of the individuals that you'd like. It's not really any ethical issues against anything like that. So what you can do is you can calculate the percentage of offspring that would result from these recombinant gametes, and what he found is that it really just depended on the distance between the 2 genes on the chromosome. If 2 genes are located very closely to each other along a chromosome, they're just less likely to have these crossing over events happening. Okay? If the 2 genes are located relatively distant from one another, they're much more likely to have this crossing over event happen. So if you look here, just as an example, okay, if you have 2 non-sister chromatids being exchanged, okay, what you can see here, if they're very close together, this crossing over event probably going to happen somewhere down here. Okay, it's just so close together that crossing over is very unlikely to happen in this small little region. So what you're going to see in these gametes is, again, it's going to look like the parents. They're not going to have any crossing over, and they're just going to be very closely linked to one another. Now, in contrast, if A and B are very distant, very far apart on the chromosome, you're much, much more likely to have these crossing over events happen. And so what you would expect from the gametes is a combination of these alleles. Okay? And based on what percentage of the offspring would look like the parents versus these potential crossing over events, you can actually map the sequence of these linked genes. And the way that they defined it was one map unit, or MU, is defined as 1% recombination between 2 genes on a chromosome. So it's a kind of a relatively, it's not a very specific term, right? Because there could be regions that are very, for whatever reason, aren't likely to have a lot of crossing over, or are likely to have a lot of crossing over, and that may affect the physical distance on a map. But this is the best that they had at the time. It was to do calculations like this. They also call these map units sometimes centimorgans after Morgan. Okay? And again, they're relative distances, not exact ones. Okay, so if you were calculating distances, just to give you an example, say in a cross, you get the following percentages of offspring. Okay? Yellow-white at 0.5%, white miniature at 34.5%, and yellow miniature at 35.4%. What order would these genes be on a chromosome? Okay, the answer is that it would go yellow, then white, then miniature. You know that yellow and white must be very close to one another because you have such a low percentage here. Okay, and what we can say is that because there's the crossing over event of 0.5% here, you can say this is about 0.5 map units. The distance between the yellow and the miniature looks like that's the biggest number here, so the miniature must be the furthest on the right. And you can say that that is about 35.4 map units. Now, if you were to subtract those two numbers, the distance between, to get the distance between the white and the miniature, you'd get 35.4 minus 0.5 at 34.9 map units. And what you can see is that is close to the actual frequency of recombination at 34.5%. Again, these are all going to be relative percentages. They're not necessarily going to be absolute numbers. Okay. So what we've talked about so far primarily are going to be single crossovers, and you can use these to determine the distance between two linked genes. But you can use these double crossovers to determine the order of 3 genes on a chromosome. So when you're studying these, you must look at 3 pairs of genes, all on the same chromosome, and the best way to do it is to study ones that are going to be heterozygous. So if you were to look here, say you're looking at genes A, B, and C, if a double crossover event happened, you couldn't really tell the difference from necessarily from the parental in some of the cases. Okay, so the non-crossover one, A, B, C, you've got it here. A, B, C, you've got this here. Now, the ones that are going to be the double crossover, if you follow it, it's going to start here, capital A. Okay, now it's going to go down, get a B, go back to this capital C, and so on with the second one. Okay, start with the lowercase a, capital B, lowercase c. If this was a single crossover event, what you would expect is probably one of them on the ends to be different from the parent, versus now you've got the one in the middle that's different from the parents. So what you can do is you can look at some of the previous research that they've done, and you can really look to see if something was a double crossover event, or if something was a single crossover event, or if there was a no crossover event happening. Okay, so when you're doing these types of experiments, one of the sexes, usually probably the female, is going to be the origin of the female gametes. The male, you're going to keep pretty consistent. And usually what you're going to do is you're going to look at a homozygous recessive for all of these traits. In this case, he was looking at the males, and so the X chromosomes here, this would be their Y chromosome. Okay? So again, going back to the female gametes, if you had no crossover events happening, you would get gametes that looked both like that female's parents. When they combined with the male, you would get basically this 1-to-1 ratio that you would expect to get. Okay? And it's very close to this expected ratio here. If you had a single crossover event, okay, you would expect it to look something like this in the gametes. You have this potential combination and that potential combination. So when they combined with this male, you can see the number of different offspring. You can look at their phenotype and see what calculations you're going to be able to get from that. If it was a single crossover event between these 2 sets of genes instead of this set of genes, you're going to get this potential offspring. Okay? And again, you can look at that observed number. If it was a double crossover event, okay, you can look and see at the combination, and you can see the expected offspring here. And what you can see in this particular case is that they were actually able to map these to the chromosome. So firstly, they knew that this must be the standard order. Okay, they were able to determine the distance between it by looking at the percentages. If you look at the overall percentage between this Y and this EC gene, okay, it's almost 5. It's 5.62. Okay, that's pretty close to the observed ratio that they're seeing of 6% for this double crossover event happening. If you're looking at the single crossover between the Y and the W, you've got a 1.5% observed that they saw, and that's really darn close. Okay, 1.56 here. In the second case, between the W and the EC, they saw 4%, and here you can see the actual map units of 4.06. Again, pretty darn close to their expected ratios from this.